All right, welcome back, everyone, from the lovely Oxford University Museum of Natural History in this sunny light here in Oxford. It was actually a while ago. All right, so we're about to start session number 7A. Remember, this is one of the split sessions. So if you're one of the ones keeping track of both, the challenge is there for you guys. This is one of the things of having an online congress, right? So cool. Remember that this session will be based on 5 15-minute talks. Actually, the talk will be 12 minutes plus 3 minutes for questions for each of the talks. Should you experience any issues with connection, remember that you always have the reconnect button at the top of the screen that you can press. Hopefully that should take care of most of the issues. And please, at any time, drop your questions. Feel free to drop your questions in the chat. And to do so, please mark the questions as such using the red circle that is integrated in the function to mark what you type as a question. All right, we might perhaps give another minute more before starting, courtesy. We have wonderful talks in this session, and we'll shortly start with the session. Please remember that we have the Discord server to meet up with people there, perhaps to tackle some ongoing projects or just catching up. We have open rooms that you can use in Discord at any time. All right, so without further ado, we'll start our the session with our first speaker, with Susana Gutarra Diaz. Susana? All right, we have Susana.
Hello.
How are you, Susana?
Thank you. Very good, thanks. Yeah,
all right, we are
to be here.
Good to have you. Presentation should be uploaded shortly. All right, here it is. Go to you.
Excellent. Thank you very much. Well, today I'm going to talk to you about a project that is part of my PhD, in which I study the hydrodynamic properties of Mesozoic marine reptiles. Tetrapods colonize aquatic habitats multiple times, and nowadays a large number of aquatic tetrapods are mammals, like pinnipeds or cetaceans. But during the Mesozoic, reptiles dominated the seas. And among those groups, the longest lived and most diverse of them were the ichthyosaurs and the sauropterygians that you can see in the tree at the bottom. Both of these groups had derived members that became pelagic and highly efficient lift-based swimmers, but they did so in 2 distinct ways. You can see at your right the ichthyosaurs as caudal oscillators with bodies very similar to cetaceans, and on the left with the 2 flavors, plesiosaurs with long and short necks as quadrupedal underwater flyers. Now, in the past, ichthyosaurs have been inferred to be faster than short-necked plesiosaurs, and this in turn faster than long-necked plesiosaurs. And these inferences were based on assumptions that the fineness ratio, that you can see on the top left, which is the length divided by the maximum diameter, can be used to infer a minimum drag in bodies. This is what is actually observed in the standard shapes you can see in the bottom left, like spheroids, for example. They have the minimum drag at a fineness ratio of 4.5. So this observation, plus the fact that many derived aquatic tetrapods, both fossil and modern ones, fall in that area, like the gray box there in the right, of, let's say, minimum drag. This led to the assumption that this fineness ratio, or 4.5 or around that, has been able to reduce the drag. And therefore, everything that's more elongated than that falls outside of this range is deemed to be draggy and swims slower. But this concept has started to be challenged in recent times. For example, last year we published a paper using computational fluid dynamics, in which we showed that the transition from at the left, you can see the transition from very early ichthyosaurs that had elongated and narrow bodies to the more derived stocky fish like ichthyosaurs did not reduce their cost of drag for a given body mass. So in fact, it's not shape, as you can see in the top chart. shape did not affect, and fineness ratio did not affect this, but what did is size, actually, that contributed to reduce the cost of drag during the evolution of these ichthyosaurs. Another recent paper from a group in Liverpool also used computational fluid dynamics to test the effect of neck morphology in plesiosaurs. They reported no significant changes of the drag coefficient due to the length of the neck. However, these comparisons do not account for volume or mass, and also drag coefficient per se is not a proxy for energy performance. So we still are not, we still don't know very well how these extreme morphologies affect the energetics of swimming. So to address this, we made a battery of three-dimensional full-body reconstructions of a diverse array of derived plesiosaurs, ichthyosaurs, and modern cetaceans using rhinoceros. And we use this to perform computational fluid dynamics in the program ANSYS Fluent. And for this, we use a protocol that had been previously validated using standard shapes with data from actual water tank experiments. So these static simulations provide a very simple model for fully submerged steady swimming. So at constant speed, thrust equals drag, and we also adjust for zero lift, and we assume neutral buoyancy. So in these conditions, we can take the drag divided by the volume as a proxy for the contribution of drag to the cost of locomotion, which is the energy that takes transporting 1 unit of mass per unit of distance. So this is the contribution of drag to that value. And when we then this is the results when we compare the bodies of these animals at the same volume. It is the plesiosaurs that produce the higher drag. And this is clearly an effect of the limbs that are shaded in darker color, because the bodies that are in lighter color in the bars do not produce these significantly different values of drag. So this means that plesiosaurs would actually need to get to produce about 30% more of energy to keep up the same speed than ichthyosaurs and cetaceans. And we didn't see that this was correlated with, so we didn't find any correlations with fineness ratio. So on the left panels, you can see with or without the limbs, this does not correlate with fineness ratio. What it does correlate with is with the surface area to volume ratio when the limbs are present, is the bottom, is the right bottom panel. Yeah, now, if we include the real sizes of these animals, so animals at their life size scale moving at the same speed, now the difference between the groups that we observed before are lost, and they are not significant anymore. In this other situation, what we see is that actually bigger is better for the mechanical cost of transport. So large animals like the humpback whale, or then the orca, followed by the Temnodontosaurus or the Thalassomedon, have very lower, have the lowest mass normalized values of drag. Now, this is an effect of the very large scaling effects that size has on the balance of drag in these streamlined animals. So we are actually dealing with streamlined forms, like you can see on the bottom, on the top right, that for these shapes, most of the drag comes from skin friction and very little from pressure drag, as opposed to bluff forms in which most of the drag is pressure. So all these animals, ichthyosaurs, cetaceans, plesiosaurs, they all behave like that. They have most of the drag coming from skin friction, up to 80 or 90%. And this is why that with bigger size, bigger means less surface area and less drag per volume of body mass. Now, the next thing we did is focusing just on the necks, and for this we computed the drag of different models with variable neck proportions, and then for the comparisons based on volume, we took the volume of the trunk, which is the place of the space of the body that contains the muscles for propulsion. So first, what we found is that that the drag coefficient at a given Reynolds number, that is same length and same speed, doesn't vary. Those are the dots. The crosses are the amount of skin friction, which is 85% more or less, which is what you would expect from streamlined shapes like this. And this does increase a little bit with the neck length, but because the pressure drag, which is the remaining part of the, which is the remaining between the two of them, gets reduced. So these two balance each other, and the overall change in total drag is negligible. However, if we compare, in the comparisons where the neck gets longer while the trunk remains a constant size, the drag relative to the volume now shows a totally different trend, which is an interesting thing. So there is no excess drag for bodies that have necks up to 2 times the length of their trunk, but for longer necks, there is indeed a substantial burden, and this increased progressively. So ratios about 3 to 4 are found in elasmosaurs, and 5 to 7 are merely hypothetical. But we do see an effect of the neck in the energy of, in the drag costs of swimming. So now, what happens if we introduce the real size to this? So for this, we have a broader sample of limbless models, different plesiosaurs, basal, derived pliosaurs or thalassophonian, polycotylids, and elasmosaurs, which cover well the diversity of body proportions and sizes. So if we compare these animals at the real scale, swimming, moving at the same speed, what we see is that the large trunks are the ones with the lowest drag that you can see on the top panel in color blue. And on the bottom panel, what you can see is that there is a strong correlation between the trunk and the relative drag per volume of trunk. So there is almost an inversely proportional correlation. And finally, what we see is that because of the large trunks of elasmosaurs, these compensate for the excess of drag of their necks. And finally, there are no significant differences between the elasmosaurs and the derived pliosaurs, the thalassophonians. Even if we remove the aristonectins, that is a subfamily of elasmosaurs that reduce their neck. So we observe how the proportions vary in a large sample of sauropterygians, which include also non-plesiosaurian forms like placodons or notosaurs. And what we see is that there is a great plasticity of necks, but most of these species fall in the area that has no impact for the energy of swimming. What we also saw is that the very large necks are only present in species with really large trunks. So about 150 centimeters or more. So these necks of more than 3 times or about 3 times the length of the trunk are associated with large bodies. And actually, estimating the ancestral estate of these of sauropterygians, we see that the ancestors of the elasmosaurs were probably large-bodied animals with maybe about 180 centimeters of trunks. So they already had quite large trunks. And the last thing we ask is whether having large trunks release somehow the constraints on the neck, and they evolved any faster relative to the other clades in elasmosaurs. And so we did an evolutionary rates of evolution analysis using the variable rates models of bias traits. And then we actually found that there is evidence for fast rates of evolution in the necks of in the neck proportions in the branch of elasmosaurs. So there you go. I packed more or less what I've been doing in recent times. And then to sum up what I just showed you, using computational fluid dynamics, we've shown that plesiosaurs generated more drag than ichthyosaurs and than cetaceans of the same mass because of their limbs and not because of their fineness ratio. We also show that this difference can be, however, overridden by body size. And then we also show that the necks can actually have an impact on the energy of steady swimming. So about 3 times or longer the size of the trunk can add substantial drag. But in elasmosaurs, the trunks also evolved to be very large, and this compensated for the potential excess of drag, and therefore we didn't find any differences between elasmosaurs and derived pliosaurs. And finally, these high rates of evolution that we observed in the neck of elasmosaurs might suggest that there is a release of the constraints on the neck proportions, that is favored by the large body sizes. And with this, I would like to thank my supervisors at the University of Bristol, my colleagues, and all the people that has helped me with collecting data, and also my funding bodies. Thank you for listening, and the Palace organization, and I will take your questions. Thank you very much.
Thanks, Susana. Yeah. That was great. So we have.
Sorry if I was speedy.
No. It's all good. So good. It was perfectly clear. And a nice presentation. All right, so time for a couple of questions. We have here, so you've explained that skin effect is lower as size increases, right? But one of us was wondering how strong is the effect of the skin, perhaps in the smaller models?
Of the skin, you mean the roughness or the texture of the skin? Yeah, that skin friction can be affected actually by the texture. So for these models, we assume that the texture of the skin is smooth because there's little evidence on the fossil record of the structures or scales. So it's probably also the very derived ichthyosaurs and plesiosaurs tended to kind of smoothen their skins. But yeah, we have to assume for these computations no differences, and we make smooth models. But yeah, this can affect.
Mm-hmm. And have your results been validated against living taxa?
Actually, in the first paper that we published on ichthyosaurs, this includes a dolphin, and dolphins have been tested in water tanks, models, scale models of dolphins made with realistic measurements, and also have been multiple times tested with computational fluid dynamics by other groups, and we have values that are consistent with these. Yeah, so.
Nice. All right. Well, there are some more questions for you, but you might want to address those.
Okay, I'll check that
as well. Please keep dropping questions for Susana in the chat, and she will answer the questions as the session goes by. Thanks so much, Susana.
Thank you very much.
We'll now invite the next speaker, who is Rachel Belben. Rachel? All right. Hi, Rachel. How are you? Can you? I don't know if we can hear your. Yeah, I think we have the same issues than before. So probably we want to do the magic that you've done before with us in the testing. I don't think we can hear you yet. Not now. Is the microphone button on, right? Yes. Okay. So you might want to try to do some tweaking. If not, it's okay. We can perhaps alter the order of the talks. And we don't seem to have audio just yet.
So their microphone's on, but we're not hearing it. Get them to close the window, log back in.
Okay.
You give some announcements.
Yeah. Okay. Let's do that then. Let's please, Rachel, try to relog into the session. That might solve the issues. And yeah. So in the meantime, I will make use of this time to remind you of some of the amazing things that can be found in our Discord server. Remember that we have the poster room online at any time there, so that you can exchange some impressions with the poster presenters. Rachel, I don't know if you've managed to close the window of the session. Exactly. Perfect. All right. Now we know that we are rebooting the whole thing. All right. So also in the Discord, there are different open rooms that you can use to meet with people, to talk about research, or to just say hi, or to basically wrap up for some of the projects. Of course, it's not possible to make up for the bar, right, for the pub, but it's all we have now in these technological times. And of course, we also have Rachel Erin, our lovely illustrator, providing us with some artistic love for all of us to see. It's going to be quite amazing how she can draw all those flash portraits in flash. I mean, at the speed of time, and the speed of light, right? So we've been enjoying all that a lot. We also have a room for PhD students in the Discord server. So hopefully Rachel will come back to the system soon. If not, what we can do is just we could move on to the next speaker. Let's just wait for another minute more. After this session, we have some time for lunch, or just we have more or less like an hour pause, and then the sessions will resume later in about an hour, sessions 8A and 8B, 2 parallel sessions. So probably because Rachel is not, okay, now we have Rachel. Nice, let's make the test again. Rachel, if you can hear us, please switch your camera and microphone on. I think we can hear now something. Okay, we have, yes, lovely. Now we have
okay.
From you.
Sorry, I'm not sure what happened there.
It's fine. It's all good. Take some extra time to catch some breath, and yeah, it's take it over.
Okay, thank you. So I'll try and be a bit speedy. My name's Rachel Belben. I'm from the University of Leicester in the UK, and I'm here to talk about pterosaur taphonomy. So taphonomy for vertebrates especially has tended to focus on single specimens. And this is often qualitative, it's descriptive and analysing admittedly very spectacular specimens such as this one here. And these specimens can provide extremely unique and interesting insights into the lives of these particular individuals. But also as qualitative data, it can be hard to compare even when you have similar specimens. And when you don't have similar specimens, it can be difficult and risky to generalise across more than across the whole group. Quantitative taphonomy, on the other hand, can provide also a potentially very powerful approach to understanding fossils. But within within vertebrate taphonomy, this has been largely neglected. So all fossils yield some kind of taphonomic data, whether this is simple counts, presence-absence data, or if it's more complex, like this posture data from exceptionally preserved pterosaurs. And compiling and analysing these data from large numbers of individuals and large data sets can, as I hope to show in this talk, yield important insights and that can match sort of the single specimen spectacular examples of fossils I showed on the previous slide. So pterosaurs I'm going to be talking about are a diverse and long-lived clade. They're Mesozoic flying reptiles. Traditionally, they're divided into 2 groups. So the first is the basal non-pterodactyloids. These are a paraphyletic group, and species tend to be small to mid-size, usually long-tailed, and they're also fairly conservative morphologically speaking. They all, with one exception, have this elongate 5th toe. So as you can see here pictured, this is made up of 2 phalanges, very elongate, very highly modified. The second group of pterosaurs, the pterodactyloids, these are much more morphologically diverse. They are anything, basically the entire range of pterosaur size, small to giant. These ones, however, had also a modified 5th toe, except it was modified in the other direction. The 5th toe is basically either a tiny little nub or it was lost entirely. And this is significant because the 5th toe is basically this was the thing that supported the uropatagium, so the wing membrane between the hind limbs that connected at the ankle. So the uropatagium is, by current consensus, and also a small handful of soft tissue evidence, in the non-pterodactyloids, the basal clade, this spans the hind limbs from ankle to ankle in current consensus, but much more patchy soft tissue evidence. The uropatagium in pterodactyloids was split down the midline, so it did not connect to the hind limbs. This is kind of dangerous thinking in the sense that this is extrapolating from not necessarily extremely concrete data onto the entire clade, and especially given that whether the hind limbs were connected by uropatagium or not probably affected the terrestrial and aerial abilities of these animals. So the extent of the wing membranes can be conceptualized by the idea of modules. So a module here is a functional and anatomical unit. It can be one limb if the limb is independent of all the other limbs. For instance, humans have 4 modules, all their limbs are independent of each other. But if the limbs are connected, for instance, by wing membrane, these modules can consist of more than one limb. So given our volant vertebrates, our birds, our bats, and our pterosaurs, birds on the left have 4 modules, as all of their limbs are functionally and anatomically independent of each other. Bats, the limbs are all connected by wing membrane, so this makes 1 module. And what of pterosaurs? So going by that current consensus, non-pterodactyloids, which the Europatagium connects the hind limbs together, is formed of 1 module in the same way as bats. In pterodactyloids, the hind limbs are separated from each other as the Europatagium is split, so there are 2 modules. And given what I was saying earlier about the slightly lack of data for the Europatagium actually being there, the extent of it, again, this is worth testing for. So I'm using quantitative taphonomy and testing these hypotheses. So the first one is that non-pterodactyloids had 1 module and pterodactyloids had 2 modules. And secondly, that the modules were universal within the groups. How am I going to test these hypotheses? So I'm going to use skeletal data and association between the limbs. So, for example, are the hind limbs more associated or more frequently associated to each other than they are to the forelimbs? How does this compare between different groups of pterosaur? And also, how does it compare across the birds, the bats, fossil birds and fossil bats as well? So I compiled basic completeness and articulation data. So articulation is composed of articulated, associated, and disarticulated, and I focused on postcranial elements, especially the long bones. I also collected quantitative pose data. So the spinal column was used as a base, and I measured the angles of the principal elements leading off it. So the humerus, the femora, the radius ulna and tibia, and the metacarpals and metatarsals. A phylogeny, this one shows another benefit of taphonomic data. So the grey box at the bottom is the pterodactyloids. The taphonomic data is highlighted in blue. So this is the data I've already collected, and hopefully I will be able to collect a bit more, fingers crossed. And the red dots show the data or the specimens where there is evidence, direct soft tissue evidence for the Europatagium. And as you can see, particularly for pterodactyloids, this is fairly severely lacking. So it emphasizes the much greater completeness of the taphonomic data than the soft tissue data. Taphonomic data also spans almost the entirety of the stratigraphic range for pterosaurs, pretty much all the principal pterosaur Lagerstätte, and a broad range of preservational types and styles. So here's a plot of completeness against articulation. As you can see, there's a fairly even spread. There's no clustering. The non-pterodactyloids aren't in one area, the pterodactyloids aren't in another area. And this is true also down to sort of the major clades group areas. This is the area, top right is the most complete and the most articulated. So these are the specimens used for the pose data. So a brief explanation of the pose data. For symmetry, I looked at symmetry down the midline of the body, the left and the right sides. So basically, if you consider the body a clock face, then the hands at 8 and 4 are perfectly symmetrical and they would score 0. Whereas the less symmetrical it goes, for instance, 9 and 8 would be very asymmetrical and score very highly. So for humerus symmetry, the bats and the non-pterodactyloids both had a correlation of they had skewed towards very high degrees or near perfect symmetry, whereas the birds and the pterodactyloids had a much more even range, even though they did skew towards high symmetry. This is even more striking in femur symmetry. So the bats and the non-pterodactyloids, again, they skewed towards perfect or near perfect symmetry in the femur, whereas birds and pterodactyloids, they had a much more even range. The poses were sort of splayed any which way. There was no constraints here, it seemed like. The femur angle, this isn't symmetry. This is basically a 0 for femur angle would be the femur pointing straight down and parallel to each other. And the higher the angle, the wider the spread. So for this, it's not quite as clear as for symmetry, but you can see that bats and non-pterodactyloids sort of skewed towards a mid to high range. And the birds and pterodactyloids, on the other hand, had again that more even and more wide spread of data. So what can we conclude for this? Firstly, that it seems that non-pterodactyloids are very closely correlated to bats, and they had so the non-pterodactyloids are very strongly supported for one module, the same as bats, as they closely correlated to, and they're also supported in the fact that they have a narrow range of postures. So this is supported by the idea that if the leg is physically held in place by a wing membrane anterior to the body by the main wing membrane, and also posterior to the body by the hind, the uropatagium between the hind limbs. So this would support that. And also that it seems, because of the narrow range, to be fairly universal, if not universal, across all non-pterodactyloids. A second conclusion is that in pterodactyloids, the pterodactyloid bauplan consisted of 2 modules, left and a right, as the limb postures are, especially the hind limbs, quite closely correlated to birds, which have independent hind limbs. The third conclusion is that although there's not much soft tissue evidence, it does correlate fairly closely to what we have, almost exactly to the results that we have. And the final conclusion is a more general one, which is basically that the study demonstrates that quantitative taphonomy can be successfully used for pterosaurs, but it also can be successfully used outside of the group. It can be widely applied and also applied between groups as well. So many thanks to Centre for Funding, University of Leicester, my supervisors, the institutions that allowed me access to their collections, and of course you, the audience, for listening. Thank you.
Thank you so much, Rachel, for that fascinating talk and bringing some taphonomy into our lives. It's always necessary, right? So, all right. So let's see. We have time for one quick question, and please keep dropping your questions in the chat. Rachel should be able to answer to those questions as the session moves forward. Let's see if we have one question here. We know, I'm going to try to read it as we go, so excuse me if I pronounce something correctly. We know that from Krasiak Beach deposits that the 5th digit of non-pterodactyloids touched substrate in digitigrade stance, which suggests that the uropotagium was not as restrictive as initially suspected. Do you think that the uropotagia was stiff or really flexible membrane?
I think it necessarily has to be flexible. Purely because, yes, they can. The track evidence suggests that, well, the track evidence proves they can walk terrestrially. Though it's flexible, I would say that even a flexible membrane is still more restrictive than no membrane at all. So, and also there is fairly clear signal in the data that suggests at least something is happening, whether or not it's a very stiff membrane. It's, yeah.
Interesting. And I actually had a question. Can those taphonomic studies be brought to the world of insects? Has that been applied to insects?
I'm not an invertebrate paleontologist, but I do actually know that I had to edit my talk slightly from SVP because invertebrate taphonomy is much more quantitative than vertebrate paleontology. So I'm sure there are similar studies, obviously not identical because the anatomy is so different. But yes.
Of course. Yeah. All right. Well, Rachel, thanks so much for your talk. That was great. Sorry about the difficulties.
No, sorry about that.
All right. Our next speaker is Valentina Rossi. Valentina? Nice.
Yeah.
Hello, how are you, Valentina?
I'm fine, thanks.
All right. Let's start your presentation in a minute. And there it is. The stage is yours.
Thank you. So hi, everybody. Thank you for being here in this session. So today I'm presenting the results of the taphonomy of soft tissues of moonfish from the Monte Bolca Lagerstätte. So first of all, what's a moonfish? So generally, we call moonfish any fish that has a laterally compressed body. And this is the case, for example, of Mene maculata, that is a fish that lives today in, for example, in the Indian Ocean. The characteristic of this type of fish is that they have long pelvic fins, and also they are silver in color with pretty evident black spots in the dorsal part of the body. And we have evidence of other species of this family, the family Menidae, in the fossil record. For example, Mene rombea from the Monte Bolca Lagerstätte. So this fossiliferous site is actually comprised several different fossiliferous deposits around the Lessini Mountains in northern Italy, and is famous for its fossil teleost fauna. However, these fossil deposits, this Lagerstätte, it's much more than that, and more than 300 species has been described so far. And this includes invertebrates and vertebrates from both marine and terrestrial environment. And also we do have evidence of plenty of plant materials. There has been some study of the taphonomy of soft tissues, especially phosphatized soft tissues in the 1990s. However, the taphonomy of other soft tissues with different mode of preservation has not been looked at so far. So this is why I starting this project, and I starting by looking at the soft tissues preservation of this specimen of Mene rombea that been discovered in 2019 from La Pescara fossiliferous site. And as you can see, preserve evidence of soft tissues, and more than 300 specimens has been found from the Monte Bolca Lagerstätte, and they all preserve, many of them, not all, preserve the evidence of eye spots, for example, or there is a dark colored material in the abdomen, and also many specimens have a striking longitudinal stripes preserved in the dorso, but those stripes are not preserved or not visible in the ventral part of the body. So the hypothesis for this project were basically to try to investigate this dark material a bit more in detail. And the first hypothesis is that these dark colored regions are melanosome-rich soft tissues, and that we have evidence of integumentary and non-integumentary melanosomes preserved in this specimen of Mene rombea. And lastly, I wanted to investigate whether, if we have melanosomes preserved, whether those melanosomes preserve evidence of chemical preservation of melanin using Raman spectroscopy. So this specimen is stored in the Natural History Museum of Verona in Italy, and at the time when I started the project, it couldn't leave the museum. So I decided to take small samples to do scanning electron microscopy and also Raman spectroscopy. So the result of the scanning electron microscopy, as you can see here in these SEM images, you have evidence of these microbodies that we can call melanosomes. And in the dorsal part of the animal, they are preserved as thin layers, and these layers are more or less actually of 5 microns, and no other structures are visible or preserved. In the eye spot, we also have evidence of melanosomes, and those are of 2 different morphologies. We have subovoid or subspherical, and also very elongated melanosomes, and those are typical of the RPE ocular tissues in vertebrates and cephalopods. In the upper abdomen, instead, we have a really thick layer of melanosomes. The thickness is almost 300 microns, and you can see the melanosomes are very well preserved. Similarly to the lower abdomen, where also melanosomes are preserved, but they're not as thick as layers are those in the upper abdomen. So because I found melanosomes in different regions, I obviously analyzed the geometry to see if there were any differences. And as you can see from the box plots and also from the little schematics in black at the bottom of the slide, you can see that there are differences in the geometry and also a little bit of the size of melanosomes in different body regions. And this is known from extant vertebrates and also fossil vertebrates, and this indicates that those melanosomes are from different tissues or organs. However, I was trying to look at the literature of modern teleosts to see if there are any evidence of this pattern in melanosomes geometry, and there were no data. So I decided to do a quick check by extracting melanosomes from a sea bass to try to see if there were any differences in geometry, and there are. In the context of the morphospace, what I can see is that the melanosomes from the Menerombea plots in the realm of the ectotherms morphospace for melanosomes, which makes sense, and they also overlap more or less with those of the melanosome from the sea bass. So we can definitely accept the first two hypotheses, that these dark-colored regions are melanosome-rich soft tissues, and we do have evidence for integumentary or integumentary melanosomes. So we can give a partial interpretation of the internal anatomy of the specimen, and we can recognize obviously the skin, which also preserve the patterns, and also we have the eye spot, and in the abdomen we can recognize two melanized soft tissues, and in particular we have the kidney in the upper abdomen, and we have probably melanosomes from heart or potentially also the liver from the lower abdomen. And those tissues are typically heavily melanized in modern teleosts and other ectotherms. So in terms of the chemistry and the chemical analysis, because we found melanosomes, we were, microbodies that resemble melanosomes, we were expecting some sort of melanin signal. So I started analyzing synthetic eumelanin to try to, familiarize with this type of data set. So here is the result. So the Raman signal of synthetic eumelanin, it basically gives the first information that it gives you is that there are two bands or two major peaks, as you can see here, and those are called D and G bands and are typical of a disordered graphite-like structure. As you can see here very briefly in the schematic, in two dimensions, the eumelanin structure can be simple, but then in 3D actually present a very heterogeneous and complex structure. So, after, in the Raman spectra, actually, there are other informations. In fact, these two bands, the G and D bands, are formed by a number of peaks that are characteristic of the vibration energy of fraction of molecules or specific bonds that form the molecular structure of your molecule or your material. So you are able to extract all the information to identify your material using Raman spectroscopy. And these interpretations that you can see here are made after a literature review to help myself basically interpreting these peaks properly. However, when you analyze melanosomes extracting from fresh tissues, the Raman peaks is not exactly the same as for the synthetic eumelanin. And this is because synthetic eumelanin has a composition pretty simple, whereas the melanosomes contain melanin that can be formed by any of these molecular monomers that you can see here in the gray box, plus there are proteins that are also present in the melanosomes. So all of those molecular fragments, monomers, and proteins can actually contribute to Raman spectrum. In fact, if you just compare the shape of the spectrum of the spectra here, they seem pretty different. In particular, and just for now, I was focusing on the position of these two peaks that are highlighted in pink, and they can be interpreted as carboxylic acid bonds that are very abundant in natural melanin samples or material. So when I passed on the analysis of the fossils, what I started with with samples from the ink sac of a Jurassic cephalopod from the UK, just to have an idea of what I expect from a fossil. And as you can see, for the Jurassic ink, but also for the Menerombea melanosome-rich soft tissues, the overall shape of the Raman spectra are very similar to synthetic eumelanin and not to natural melanosomes. In particular, also in the melanosomes from the integument of the Menerombea, it seems to be, as you can see from the arrow pointing at an extra peak that seems to be present only in the integument and not in other samples. However, I'm still in the process of interpreting the Raman peaks So I don't have a conclusive answer for now for this. It's just, it's there. So to try to interrogate this this data set a little bit more, I did a preliminary PCA analysis just to see what it would look like. And basically, this PCA is telling me that the fossil samples are more similar to each other, and also that fossil melanosomes are different chemically from modern melanosomes extracting from a sea bass. And these differences from this plot is basically driven by the absence of carboxylic acid bonds in the fossils that seems to be present, if my interpretation is correct, in the modern melanosomes. The loss of this particular bond in the molecular of eumelanin is known and is being observed after maturation experiments, and this work has been done by Ito and colleagues, and it's been published in 2013. And so it is expected that those bonds are not present in fossil melanosome samples. So basically, to come back to the third and last hypothesis, Raman spectroscopy is not totally conclusive for now. We might have evidence of the genetically altered eumelanin. However, I have to do a little bit more analysis and potentially experiments to constrain this result a little bit better. So in conclusion, the integumentary pattern of Menerombea is different from that of the species, not genus, Mene maculata. Longitudinal stripes indicate specific behavior, for example, shoaling. We now have evidence of non-integumentary melanosomes in a fossil Teleost, and this adds in the number of specimens that we were able to identify non-integumentary melanosomes in the fossil record. And as I said before, Raman spectroscopy is not totally conclusive for now, and further analysis will potentially inform of the chemical taphonomy or molecular taphonomy of melanin in fossils. And with this, I finish my talk, and I thank you all for listening, and I would like to thank the Italian Paleontological Society for giving me support to do this project.
Thank you so much, Valentina, for that really interesting talk and telling us about the intricacies of melanin and melanosomes. Yeah. We have some time for questions, so please look in the chat. There are some specific ones that you might want to address in the chat, perhaps. That might be better. I was actually surprised, right? Because getting into this non-integumentary melanin, I might have missed that because I've been coming here around. But why are those tissues melanized in the first place?
So this is a great question. So in modern taxa, there is a little bit of study on this topic, but I feel like it's still very patchy, the publications. So one theory is basically that internal melanin is kind of helping the system to, for example, binding metals that could be dangerous for the cells. And also, so this metal homeostasis is obviously very important. Plus, for ectotherms, so for amphibians, reptiles, and fish, it seems to have an important role for immunity. So that's another hypothesis that has been basically kind of like put it out there from other work that has been done. It's very interesting because not every animal that is alive seems to have internal melanin. There is a group of people that are actually studying this, and they suggest that there might be a secondary acquisition through time, but also there is secondary loss. So there is a lot of work to do, and I feel like that paleontology could give us a really nice perspective, looking at the fossil record.
Nice. Another question popping in the chat for you. What is the mineralogical composition of the fossil and microbodies? Any elemental EDX data on them?
So I've done some very quick EDS, to be fair, and it wasn't very conclusive. So I mean the Bolca sediments are limestones. So there was loads of calcium signal. There is also a little bit of magnesium. So that's what I got from the EDS. I would like to do more like metal analysis, metal chemistry analysis to see if there's any chemical signal from the melanosomes that is potentially different from the sediment. But that's in the future.
Of course. Yeah, nice. Thanks so much. You have some few more questions in the chat, but please feel free to answer to them if you can during the rest of the session. And thank you thank you everyone for providing questions as well. Thanks, Valentina. Thank you.